So it's a pleasure to be here and an honor for me to be talking to you. So the talk today is to kind of provide an update on a talk I gave just over 2 years ago to the Vancouver Prostate Cancer Support Group. And my goal today was to, a little bit of repetition is always good, to explain a little bit what radioligand therapy is for those of you who may not have been there at the prior talk, but as well would like to ask questions about what it is. If you put me in a hardware store trying to find out the names of tools in English when I grew up in French, I'm completely lost. So the language is sometimes different. So please don't hesitate to ask any questions, because I live in my own specialized world. So it's easy to come up with terms that may not mean a lot to people outside the field. So there's only good questions. So, and I'll try to leave as much time as possible for questions when we get to there. So before I start, this is something we have to do, and it's important for us to disclose whenever we give talk about material that are related to our research. It's about conflict of interest, because part of our job of researchers is discovering new treatments, but it's also to take them to the clinic. And unfortunately, we can't rely only on government funds to take a drug or new developments or new techniques all the way into making them accessible to people. Commercialization and working with businesses is the way we have to go to make things accessible, not only in BC or Canada, but elsewhere as well. So it's part of our role as a researcher. So we always end up in these situations where we have patents and we might stand to gain financial benefits from the work of our research. And part of the way is to manage it properly and disclose it properly as well. So there's a number of patents and things that I'll be presenting and showing you include some of that work. Where the policy at the university and at BC Cancer is that when there's revenues coming in, the expenses that led to this patent are deducted, but then the revenues are shared between the inventors and BC Cancer and the institution. So as part of that, we did found a company a couple of years ago called Alpha 9 Theranostics, and that company just got venture capital investments in it. It's a spin-off company from UBC and BC Cancer. I'm one of the 3 co-founders, but obviously now as money comes in to finance the company, our stake in the company gets progressively lower. But some of the work that we've done in the lab is going the commercial route. So I just wanted to be clear about this. What happens is BC Cancer always retains the right to use it for its own patients. So there's, it doesn't pay royalties or anything when it uses it for its own purposes. So prostate cancer in BC, and that statistic is telling. I was just looking up what the estimated numbers were for last year. And when I presented the last time, it was early 2019. It was reporting the numbers for 2018. And it was 60 men in BC with prostate cancer. That number is now 3,500. And this is caused by 2 factors: population aging and the population growth in BC. And we're hitting typically a fairly significant increase in the number of cases. A tsunami of cancer cases are coming our way, and it's getting worse as time goes by over the next 20, 30 years. We didn't have the statistics compiled for the number of people who died from the disease, but it's a very sizable number of men who pass away, unfortunately, from this disease. However, the cure rate is high. So 1 in 9 men will develop prostate cancer in their lifetime. 1 in 29 is expected to die from the disease. And our goal is really to reduce that number as much as we can. Most men who get prostate cancer, as you know, are above 60, but we all know guys who are in their 40s or 50s who've been affected by the disease as well. So with the primary treatments, I won't go into too many details. I'll spend a bit more time talking about some of the new treatments that are coming. But currently, the main problem we have, there are 2 main issues. One is that typically some men have quite a few side effects coming from the androgen deprivation therapy. And probably many of you knew this way better than I do, but the fatigue, the lack of energy, and it varies among people. Not everyone has the same level. Some people have few symptoms, some are really, really inconvenient. So people are looking for alternative approaches to treat their disease and delay the need to get this treatment as much as possible. But there's also the problem that happens when the cancer becomes resistant to those treatments. And then there are a few drugs and they work, they're effective, but they don't work long term. And once it reaches that stage, it's very, very hard to cure prostate cancer because a prostate cancer typically develops and it becomes resistant to different types of treatments. So there are some drugs like abiraterone, enzalutamide, apalutamide that can control the disease. The response duration is very variable among people affected by cancer. Once the cancer progresses under those drugs, then it's chemotherapy. And chemotherapy is docetaxel chemotherapy. This is the agent that's most commonly used. When my colleague, Dr. Kim Chi, looked at this the last time, only half of men who would be eligible to receive chemotherapy actually do receive it in Canada for various reasons. Either because they have other illnesses that make them not so great candidates for chemotherapy, or people choose not to go to do it because of the undesirable effects with chemotherapy. So there's a big unmet need at finding treatments that are better tolerated and work as well or better than chemotherapy. And this is where nuclear medicine comes in. So nuclear medicine in a nutshell is we use radioactive drugs. And I commonly joke that we inject it in people and see what it does. But in reality, we use this both for diagnostic purposes, meaning we use the radioactive drug to image how the organs work, or we can use it to detect cancer as well. But we can also use it to deliver a therapeutic payload, meaning instead of using radioactive isotopes that emit rays that escape the body for which we can make images, we use a radioactive drug to deliver a treatment dose of radiation to the cancers. So it traditionally was used, nuclear medicine was used to study organ physiology, study the biochemistry of the body in vivo, in a living individual. But now it can target specific abnormalities that are present on cancer cells, but are not present at the surface of normal cells or elsewhere in our body. And typically, we can do images by using traditional nuclear medicine techniques. So this is an example. The image you see there is a classical bone scan. So bone scans are commonly used to find whether the cancer has spread. So this particular individual, you see the dark spots in the ribs are caused by the cancer spreading. And we led a multi-center Canadian trial comparing a traditional bone scan with a new type of bone scan done with a PET scanner. And those scanners provide much sharper images. We can detect more lesions. But the question with that trial was to see, not only see if we saw more lesions, because it doesn't matter if you've got 7 or if you've got 9, for example, in most cases. What matters is whether the cancer is spread or not. And we were able to show with that study that this was about 10 to 15% more accurate than a traditional bone scan. So we can expect to see some practice changes and a shift towards using a PET scan even for bone scintigraphy. What's happened in the meantime is that there's much better things that have been happening. So first of all, the PET scanners are also progressing quite a bit. Now we have machines that can cover an entire body from head to toe, and we can image what happens to a radioactive isotope in the entire body within literally seconds. So we can do things such as this image, which we can image in real time the entire body over a period of time. But the technology now exists that we can do a head-to-toe PET scan in about 30 seconds instead of the current 30 minutes that it takes, 20, 30 minutes with current machines. So this is a US group led by a physicist called Simon Sherry that developed the first prototype. Now we're seeing a lot of improvements in PET scan technology for prostate cancer detection, for example. There's also been quite a bit of progress in our ability to make molecules that will very specifically home in to the cancer. This is progress of new radioisotopes, new chemistry techniques to make those molecules and make those radioactive drugs more effective. Now, in order to treat cancer, we don't use the same radioactive isotopes as what we use to diagnose them. So we will use therapeutic radioisotopes, and there's a couple that I'm certain have heard about. One is called Lutetium-177, and the other one is Actinium-225. Now, what this picture shows is a small atom. And you have in green, for example, are protons, and in red are neutrons. So these are kind of the small elements that are lumped together to create an atom. What happens, and then obviously we've got billions and billions of atoms, even in a small grain of sand, for example. What happens with radioactive, a reason a radioisotope is unstable is because it's got too much energy. And in order to become stable, it needs to find some release. It's kind of having a kettle or that's boiling. At some point, it needs to release steam, otherwise the pressure just builds up. And the way the steam that it releases are particles. So those particles can be light particles, and these are called beta particles. These are small projectiles that go off, are emitted by the radioactive isotope, and they'll knock down. It's a little bit like throwing a golf ball. They'll cover a certain distance, but they'll knock things on their way. And there's also some radioactive isotopes that are much more difficult to make, and they're rarer, such as Actinium-225, but they emit much larger particles. So those particles cause a lot more damage, but it's like throwing a bowling ball. They don't go as far, so they don't cause damage far away from where you are. So when you break a window, people will know who it is who threw it. But it's much more damaging to the cancer cells, and it doesn't damage healthy cells quite as much. So this is the difference between what's called beta emitters and alpha emitters. And you'll see both are making big inroads in prostate cancer therapy, both with lutetium and actinium. So the way we do this is we take a drug, we attach the radioactive isotope to the drug homes into the cancer. Whatever doesn't go into the cancer gets eliminated. So typically it'll go through the kidneys and the bladder, and you flush it down the toilet, and in Vancouver it would go down the Fraser River, but by the time it gets there, it will have spontaneously degraded into stable isotopes, which are not toxic to the environment. Now, the way we use these such drugs in nuclear medicine is, first of all, we know what we're looking for. So we have a specific cancer target. In the case of prostate cancer, there's a few targets that have been identified that are present on cancer cells, but not present elsewhere. And it's actually why we're seeing much more advances in prostate cancer compared to, for example, breast cancer, where there's fewer of those very specific targets that have been identified. And then what we do is we do an imaging test, and those tests are both used to localize disease and make sure things are, so we know where the cancer is spread. So it's commonly used for diagnostic purposes. So a PSMA PET scan, for example, is really used to identify where the prostate cancer is. Is it localized? Is it at multiple sites? And that guides treatment. But it's also used to confirm whether the target for the drug is present or not. And if it's present, and if the imaging test is positive, then people can be treated by the treatment version of this drug. And those who are negative, then we need to find other things to treat the cancer. In the case of prostate cancer, the vast majority are positive, for example, for the PSMA protein, which we're targeting. And then people are amenable to be treated by this treatment called radioligand therapy. So the ligand means this is the targeting drug. Radio means because it's using a radioactive isotope. So you'll see a series of different names. There's no real standard on how to call this. Some people call it endoradiotherapy, meaning it's internal radiation therapy. Other people call it radioisotope therapy. So and then when we target other cancers, it's also called PRRT. So you may see all of those names. When people use alpha emitters, they call it targeted alpha emitter therapy, TAT. But essentially, radioligand therapy encompasses all of those. Now, this became very popular for a type of cancer, an uncommon type of cancer called neuroendocrine tumors. And those tumors express a very specific abnormality called the somatostatin receptor, which is a little bit like the PSMA protein for prostate cancer. We have homing molecules that target in there, and it's actually much better for imaging than any other test that we have, including conventional PET scans. And this is an example comparing a PET scan where we look at how much sugar the cancer is using, and neuroendocrine tumors like prostate cancers often don't use a lot of sugar. But when we use a specific imaging agent, then we can start to see a lot of tumors that we could not see before. So the dark spot that I'm pointing with the arrow are actually tumor sites. What you see here are the kidneys where the drug is getting excreted. What you're seeing is the urine, and that particular drug binds to the normal prostate. So this is why you see that dark spot there. This is not something we're trying to do. It's just kind of an area where that particular drug binds to. So what's changed is that in late 2017, there was a major landmark paper published in the most prestigious journal in medicine called the New England Journal of Medicine, which showed that if you compared standard treatment with radioligand therapy with this compound for neuroendocrine tumors, the tumors progressed at a much, much slower rate. People lived much longer, and it was very, very significant. So this changed the whole field, because what was a fringe therapy that people were kind of skeptical about, uses radioactive isotope, it's going to be complicated, it's costly. Now, all of a sudden, the world's attention were brought to, man, this works, because there was hard, solid evidence that showed that this worked. And this led a company like Novartis has invested so far in $6 billion to acquire multiple companies to establish a presence in the field. And they're massively investing in research and development in the area. The other thing that's happened as well is obviously prostate cancer. There's fantastic data coming out, and I'll talk a little bit about this in the next few minutes. So since I last gave my talk, since this was an update, since 2019, what's happened is that Lutathera, which is this drug here, the brand name is Lutathera, has received approval by the US FDA and Health Canada. And what this means is that the infrastructure to deliver those treatments is being implemented everywhere to start treating patients with neuroendocrine tumors. And this sets the stage for patients with prostate cancer to have access to these treatments. So there's still some issues about funding. This is a very expensive treatment, but this has been approved by Health Canada. In BC, we're reimbursing as well. Most Canadian provinces are starting to offer this for neuroendocrine tumors. In terms of prostate cancer, and I'll review briefly what they are, there are two very high quality prospective clinical trials that came out of a group in Australia that were published showing the efficacy of this treatment for prostate cancer. And that has influenced quite a bit the field to move forward. There's been a lot of articles published on PSMA targeted treatments, mostly with the radioactive versions, but there's also non-radioactive treatments being developed. And what's happened recently is that there's a phase 3 clinical trial called the Vision trial. We participated in this in Vancouver. This is a drug company-led trial. It was completed last year, and now they just announced last month in March, well, a little bit more than a month ago, in March 2021, that the clinical trial had met its objectives. So this means that this drug will be approved by the FDA and Health Canada. It'll probably take a year or two for approval, and then usually it takes a year or two for the provinces to agree to evaluate the cost benefits and the health economics and start planning the implementation in the province. So this is definitely coming, in my opinion. So now these treatments target a protein called the prostate-specific membrane antigen. So this is a picture of the protein in the upper right corner. So the proteins are some of the building blocks of life. They're present in every component of our organism. Every cell is coated by different proteins. And this particular protein is present in the abnormal cancer cells in fairly high amounts. And typically, the higher amount, the more aggressive the prostate cancer is. And when prostate cancers become unresponsive to the drugs that block the androgen hormones, the male hormones, the PSMA protein gets expressed at a higher level. Despite this protein being extensively studied, we don't really know why it's expressed in prostate cancer, what's its function, or how it helps prostate cancers grow. It was extensively studied for its role in brain damage because when people have stroke, it releases some toxic substances in the brain. So people had developed drugs to block this enzyme. This is an enzyme. It converts one chemical compound into a separate chemical compound. So people decided to take those drugs and attach a radioactive isotope, initially to study the brain, but then people realized that this could be good for prostate cancer and started using it. And these are some examples of some drugs, and these are pictures from here in BC of people who are participating in some of our imaging studies. This is the first mainstream compound, and this was developed in Heidelberg, published initially in 2009, the first images of patients in 2013. They were published, the first pictures, I saw them in 2014, 2015. So this is a small molecule that binds to the abnormal protein, and this is what carries a radioactive isotope. And all of the PSMA targeting have a similar kind of related structure. And this is an example of somebody who has a new prostate cancer. This is a prostate cancer just below the bladder. Sometimes when we want to take better pictures around the bladder, we give a diuretic. A diuretic increases urine flow through the kidneys. So it's a bit uncomfortable because the bladder fills up quite quickly. And we have to make sure people don't have to empty their bladder where they're in the middle of the scan, doing a scan, because we don't want them to get up while we're doing the scan. But essentially it dilutes the activity that gets excreted, and we can see the lesions there. All of the small dark spots here are prostate cancer metastases that have spread. This can detect tiny, tiny lesions as small as 3 millimeters, 2-3 millimeters. We can find them with this scan. And this is another example of another trial we're doing. So we brought both compounds, the gallium and the F18 compound, to BC. Currently, we're scanning only in Vancouver. We're trying to expand that elsewhere in the province. But the difference is that some drug companies, because this one came in earlier, people require a scan using this compound to be eligible for clinical trials. So we didn't want to leave out our patients from participating in some of those trials. So we brought this compound as well. And this is what we mostly use to diagnose prostate cancer. This compound was developed at Hopkins. My own lab was developing a series of other compounds, but because we felt we could get this faster to the clinic, we introduced it and brought it to an open label trial. Now, I was expecting to receive 2-3 requests for people to participate in that trial per week. We're doing 10 per week now, and we're still not meeting demand. So this has been hugely popular because it can spot when the PSA rises, when the hormone, the blood marker rises. We can readily detect and quite quickly. In about 93-94% of patients, we're able to figure out where it's coming from. There are some patients who have negative scans despite the fact that we know the prostate cancer is coming back. And maybe I'll discuss a little bit about this. So now this imaging agent completed a phase 3 trial. There's a company that picked up the license from Hopkins and is commercializing that. This will likely be available at some point. We're still making this compound available through open label trials in British Columbia, meaning people still have access. But the infrastructure, the speed at which we're able to expand the manpower, get our budgets and so on to provide a service is outpaced still by clinical demand currently. We're working hard to improve this. I'll quickly jump over this. I have a little bit of duplication. So this is just the first 200 subjects that we scanned in BC. We analyzed what was the detection rate based on the PSA level. And the higher the PSA, the easier it is for us to localize where it's coming from. But even people with fairly low PSA, we're about 70% detection rate. When it's above 2, for example, we're about 95, 96%. So it's really an excellent scan to detect where the lesion is. And this is not the experience from somewhere else. This is really our experience in BC when we evaluated that. Now we've done scans on over 1,100 people in British Columbia so far. I think we're probably the busiest group in the country currently. These are some examples of how it can be used to detect prostate cancer. This is an example of prostate cancer coming back at the top of the prostate gland. This person was treated by brachytherapy. This is helping us also figuring out what are the patterns of recurrence, and we can feed that information back to try to improve the treatments, as they're delivered. This is the effect of what this has had in the patients who've received it. So we looked at people who had these PSMA scans, and when surgeons or radiation oncologists treated them, what happened to these people? Now, the problem is that most often when the disease comes back, the standard treatment would be to treat people with drugs as well at the same time, not just give radiation or surgery, but combine that with androgen deprivation. So about 90% of people get both treatments. So now we don't know really, is it because my scan was correct and localized the only site of disease, or is it because the androgen deprivation is just so effective? And it'll take us years and people who get both years to figure out whether it was effective or not. So we only looked at people who could not get the androgen deprivation or would not take it. So there are two groups of people, for example, who have heart disease, and they would recommend not to get androgen deprivation because it would increase the risk of heart disease progression, or people who refuse to take it because they don't like the side effects. So we only looked at people who had just local treatment based on the disease. And as you can see, the vast majority of people respond very well, but there's microscopic disease elsewhere that we can't pick up. So there's people who seem to be very long-term responders, but there's a group of people in whom the disease may still come back despite using radiotherapy and surgery. And what happens then is that the disease progresses, as in this example. This is someone who had a unusual site of disease. They could not treat that by radiation. They decided to wait and see whether this was a real finding. Rescan a few months later, the PSA was progressing, and then you can see the cancer is progressing. So obviously, when the local treatments don't work, and we know that they won't work 100% of the time, in this case, when it's relapsing, somewhere between 30 and 50% of people will have long-lasting control, and we're going to update this data as it comes in. But we need to offer treatments with this, and this is where the treatments come in. And this was started in Germany. They bypassed all of the standard mechanism for clinical trials. Normally, it would take a lot longer and more rigorous approaches to do that. But they use what's called a compassionate use, which is allowed in Germany, meaning you could take a non-approved drug and start treating people with severe illness right away. And there's pros and cons of using this. The cons is that you don't get high quality data. It's very hard to figure out whether the treatments are really effective and how long or not. The pros is that it makes things accessible much faster. And we've learned a lot from their experience. This is an example, all of those dark spots, except the kidneys here and the salivary gland and lacrimal gland. So this is what makes tears and saliva. All of the other spots is where the prostate cancer is spread. And you can see from the baseline treatment over the course of 4 treatments, and this gentleman had a fantastic response. The PSA went from 387 to just above 1. And there's a number of people who are extreme responders who respond super well to this kind of treatment. There's a lot to learn still to figure out why some people respond and why some other people do not respond. But what's changed are these 2 studies. When I last spoke, the study results were available. This was a study that eventually was expanded to 50 people, where they did a really well-designed clinical trial to assess how this treatment worked. And you can see that there's a few people that are not, the bottom curve, what you're seeing there is that 0 is the baseline, the PSA which they started. And when it's above the curve, means after receiving the treatment, this particular individual did not respond, but all of the others had a decline in their PSA level following treatment. And some of them a very, very significant response. Those in blue is the PSA decreased by more than half. So this is the best response they saw in their blood level after being treated by this drug. So the side effects were mostly dry mouth, as well as a decrease in blood count, which was reversible. So that happens when the cancer is spread to the bones quite a bit, and the bone marrow, which produces our cells, get affected. Now, this is what happens. This is a new study that was just published. The same group in Australia. They have a fantastic multi-center group. They're able to set up trials and get those trials underway very quickly now, thanks to Dr. Michael Hoffman, who's a colleague of mine, and a group of other investigators. And this time they took 200 patients and they randomized them to receive either the radioligand therapy or be treated by what's called a second-line chemotherapy. So this is really the last chemo that oncologists will consider giving. There's another chemo called cisplatin, but they typically will give that treatment until they will tell people that there's nothing else they can offer. So they compared the two, and this is the response to the chemo. And as you can see, the chemotherapy is still quite effective. It decreased more than about 37% of patients had a PSA decline of 50% or more. But when they gave the radioligand therapy, that percentage was 66%. So it was much better than that type of chemo. And they looked at the progression-free survival. That means how the proportion, the percentage of patients that do not see their cancer progressing. So this is not the overall survival. But as you can see, the disease still progresses, but it progresses slower with the radioligand therapy compared to the chemotherapy. So this is great news because it means that this treatment is better than the chemo, the last line of chemo. The other things they found is that this treatment gave much fewer side effects. It was much better tolerated by patients. And I know people who've had this treatment. I've treated patients. They'll be in the same afternoon. They'll just be up on their feet and happy to go shopping. One gentleman a few years ago was telling me he went to Germany to get the treatment. In the morning, he received his treatment. In the afternoon, he was hiking in the mountains, exploring ruins. So the treatments are not without side effects, but they're better tolerated than chemo for sure. So what the other big trial was this big study that was conducted worldwide with 750 men. We participated in this study, and several people in BC were able to access that trial. So this was really a little bit more like this, the phase 3 trial, so that the definitive evidence of people who had received chemotherapy who were looking at options. And this trial was completed. 2 out of 3 men were receiving the chemotherapy, the radioligand therapy. The other were treated by best standard of care. And the results of the phase 3 trial were just recently announced. So that means that the radioligand therapy was more effective than best standard of care, which could have been last lines of chemo or other supportive care methods. Now in BC, we've been working quite hard to bring in another study, another randomized trial. This is a trial that is funded by Prostate Cancer Canada, now part of the Canadian Cancer Society, and funded by the Movember Foundation behind Prostate Cancer Canada. And this study is really comparing with the standard chemotherapy. So the first line chemotherapy we would give when people progress after a drug called abiraterone or enzalutamide. So this study randomizes people 1-to-1. So the first arm is getting the radioligand therapy, the second arm is getting the standard chemotherapy, and see which one is working better. So in order for people to be eligible, they cannot have received chemotherapy in the castration-resistant setting. They might have received it in the early diagnosis. If, for example, somebody presents with metastasis at the onset. So this is really treating earlier than what most people would typically treat with radioligand therapy. So this is before chemotherapy rather than waiting after. What we're trying to find out is it better than docetaxel and is it better tolerated? And what happens is we're not leaving anybody out to dry. So there's the 2 arms that people progress under chemotherapy. Then upon progression, they're allowed to cross over to the other arm and receive the radioligand therapy. Same thing if they progress under this, then they can receive chemotherapy. And we'll learn about as well. The other big question is which one should we give first? Should we start with chemo and give this treatment? Or should we start with the radioligand therapy and keep chemo as the last line of defense? And then we're going to look at which patients are progressing and so on, and the adverse events, the quality of life, as well as measure how many hospitalization and so on. So we can do a very important component of for public acceptance, which is how much does it cost and what are the cost benefits of doing one approach versus another? Because cost ultimately comes in. You can imagine with thousands of men needing these treatments in Canada, if it costs, let's assume $200,000 for every treatment, that means, could be a very significant cost in the hundreds of millions of dollars per year. So the governments want to make sure they get value for money. And value is typically defined not by government, it's defined by the people. How much do you value improvements in quality of life? How much do you value extension of life and so on? So this is going to be part of this study as well. So where are we with this study? So we partnered with an organization called the Canadian Cancer Trial Group. So they're used to run multi-center clinical trials across Canada. This is a nonprofit, publicly funded group. Every major cancer center participates and is part of that group. They're based in Kingston, their main office, and they're very meticulous in the way they do this. So it took several months for them to revise and review the protocol. We finally got Health Canada approval in late December 2021. Research Ethics Board approval was received at BC Cancer in late February, and we should open up this month and get our first patients in as the first Canadian sites. We expect Montreal will probably be the second site in Canada, and Toronto shortly thereafter. Maybe not Toronto, it might be Hamilton. So that study, we're very excited because it'll change practice. It doesn't overlap with what's been done before. We'll learn a lot and we'll learn something new. What else is happening? There's still a lot of what's called compassionate use trials or just compassionate use practice. And they're all switching from this drug, which Novartis has acquired the patent, but didn't want to see people using it outside of the approved indication. So the supply of the precursor for that drug has dried off for researchers who want to use it for compassionate use trial. And this has affected us significantly because we had done all of the manufacturing process set up. We were ready to go with the clinical trial when Novartis acquired the previous company Endoside, and they changed their policies not to make this available anymore. So there's another compound out of Germany that, the patent situation is a little bit less clear. So a lot of groups have switched to this one. The problem is that it's just not quite as good as the PSMA 617. It works, but it's not quite as good. There's a Canadian company called Point Biopharma that has acquired a German patent, and they have a compound they call PNT 2002. And they're just initiating a trial called the SPLASH trial in Canada, US. I don't know if they're running in Europe. And what they're doing is not comparing it with chemotherapy. They're comparing it even earlier against abiraterone and enzalutamide. So their hope is that this will work better and might be pushed earlier in the treatment paradigm. This compound, I don't know the details of what it is, but from everything I know, is very similar to PSMA INT. They're pretty much similar, so likely to be effective. So this is good because we're going to see competition in the marketplace, which should keep prices down, which would improve accessibility. Now, there's a lot of other trials going on now with PSMA 617 and PSMA INT. And what's interesting that I wanted to point out was now there's combination therapy trials, combining it with different drugs. So for example, olaparib is a drug that's starting to be proven effective in a form of prostate cancer that's linked with an abnormal gene that has also been associated with breast cancer called the BRCA genes. So people who have family mutations in this are more likely to get prostate cancers. Olaparib is effective in this, but it also prevents cancers from repairing damage caused by radiation. So the combination of this with olaparib could prove more effective, but it also could prove more toxic. So there's interesting studies going on to see the combination. There's also combination with a drug that's used to activate the immune systems to treat cancers. What's happening, the rationale is when we damage a cancer cell, it activates the immune response as well. And so combining both might rope in the immune system to support the response that people are experiencing with treatment. And that will be very interesting too. There's also combination with androgen deprivation therapy, combination with enzalutamide. So we're going to see a lot of combination studies now to try to improve on the efficacy of PSMA 617. There's also been breakthroughs, and I did show this the last time around, is that if you switch the isotope from actinium, if you recall, this is the one that has the bigger particles. and is felt to be more effective. If you switch to lutetium, like this gentleman, and this has made, everybody shows these images to show how potent actinium is. This is a patient who had quite severe advanced prostate cancer. They tried 2 treatments with lutetium. Treatments were not working. So they switched to actinium, and then there's this massive response to the point where there are some people who are brought in remission by the use of actinium. Very few patients, though. The majority still will have a partial response, and then eventually it comes back. So there's ways we need to improve further on those treatments. But I wanted to use this image to point out something important. If you look at the dots there, these are the lacrimal glands that produce tears, and these are the salivary glands that produce saliva. And if you look at the images over time, you can see that you don't see those glands so much because the compound accumulates, but we don't want it to accumulate there, but it's kind of a side effect. It's an unwanted area of effect where it accumulates in fairly high quantity and also accumulates in the kidney. Nearly 3/4 of patients who are treated by actinium need to stop the treatment, even if their cancer is responding, because they get severe dry mouth to the point where they have a hard time eating and so on. So this is a major concern with the treatment because it makes people not being able to complete the treatment. The other concern is the kidney, and that's true for both lutetium and actinium. When you give those, because it sticks in the kidneys, you can damage the kidney, and there's an upper limit of how much radiation we can give to the kidneys before they shut down. So this kind of puts a cap on how much of those radioactive drugs we can give on a lifetime basis to an individual. So we can't go too high. So that means that we can't improve the efficacy without damaging the kidneys with the current compounds. So there's kind of an upper limit of what we can do with PSMA 617 because of these undesirable side effects. Whether it's actinium or lutetium, there are similar constraints, although actinium is quite potent. Now, I was quite interested about this in part because there are people who've used antibodies that have been tagged with radioactive isotopes, and those antibodies target PSMA. And when you use those antibodies in people, you do not see the salivary gland. So to me, that indicated that this is not the PSMA protein that's present there. It's something else that's accumulating the compound. And I'll very quickly go to a picture of the compound to explain where the thinking was. So this part of the molecule here, not the one in red, but the one on the right-hand side, this is an amino acid that's present in any food we eat. This is called glutamate, and it's attached to the rest of other chemical structure, but it's very important for PSMA binding to have this glutamate there. So I started wondering why we were seeing this salivary gland accumulation. So, glutamate, it's also called, when you put it as a food condiment, it's called MSG, monosodium glutamate. So a lot of traditional Chinese food would add glutamate to preparation to improve the taste, because it kind of adds a meaty taste to it. This is why you see no MSG, because it was associated in the 1970s with high blood pressure and headaches, which it had been debunked that it actually is caused by glutamate since then. But anyway, a physician wrote a letter saying his patients were getting headaches after going to a Chinese restaurant, and it was unrelated to glutamate. But I thought, since it's such a potent stimulator of salivary gland flow, after biting a piece of lemon, after lemon, MSG is the most potent stimulator of salivary flow. So maybe there's something in the salivary gland that is triggered by the glutamate portion of this molecule. So we gave mice high doses of MSG, the same MSG that is used as a food additive, and we noticed that we could significantly decrease salivary gland accumulation without affecting tumor uptake. So we gave mice quite a bit of MSG, and the ones on the left is a picture of a mouse with PSMA 11, so that's a gallium-labeled compound. You see here prostate cancer. These are the kidneys. So this tracer accumulates in the kidneys, but these are the salivary glands. So when we give a lot of MSG, salivary gland went away, tumors stayed there, but everything cleared faster, including the kidney. So we saw a nice decrease in salivary gland and kidneys. So I said, I found the solution. So I decided, well, we'll just give people a bunch of MSG and we can reduce these unwanted side effects. So we did a trial, and a number of volunteers to whom we're very grateful were willing to get 2 PSMA scans, one without and one with MSG. So we gave the largest amount of MSG we could possibly give. We mixed it in with tomato juice, and we gave people one visit was regular tomato juice with salt. The other was half salt tomato juice with MSG, just so we can kind of mix the taste. Most people could figure out which was which because the tomato juice with MSG was tasting much better. But so, and I was really, really hopeful we'd see a decrease. So we did want to use treatments with this. So we used a diagnostic scan. And the bottom line is what happened is that we did see a significant decrease in the salivary glands and kidneys. And you can see these graphs just show the decrease in all of the unwanted organs, but it also decreased tumor uptake about the same level. So unfortunately, this approach, the MSG, what we've shown also affects how much of the tracer gets accumulated or the radioactive drug goes into prostate cancers. So this was really not an effective approach. So we've decided that despite the hopes not to pursue this. Now we were developing in the meantime, better imaging drugs and better radio-labeled, better drugs for imaging, in part because I'm a little worried that once it goes into a patented compound by a pharmaceutical company, they will break the bank in BC to do thousands of scans per year. So if we developed our own, then BC would get access to free imaging and potentially therapeutic compounds. And we came up, and this is the image of a mouse. I typically don't like to show mouse images, but we have to go through mouse studies in order to see if those things work or not before going to patients. And we were able to find a new class of compounds that have no kidney accumulation. So this is just after 3 hours. You see the kidneys where the dotted arrow is are completely gone. And we saw absolutely no salivary gland accumulation. And we fiddled with several parts of the molecules to detect this. And beyond that, these compounds also have better accumulation in the prostate cancers. We developed diagnostic versions with both gallium and fluorine-18. So what we saw here is an increase in tumor uptake, decrease in kidney and salivary gland uptake to up to about 90%. So we're quite excited about this because we've got better imaging agents now. And this one here is coming into clinical trial next month. So we've done all the work to take this to the clinic. But we also thought, you know what, we'll just try to make better therapeutic compounds at the same time because we'd gained a lot of experience playing with those molecules. And this is where we are now. And if you recall, a couple of years ago, I asked, would you better get something not quite as good faster or get something much better and take the time to develop this? And this is an image with PSMA 617 where you see the kidney uptake and the tumor uptake. And this is the new compound we've developed. This is with lutetium. We also have the actinium version where we've literally multiplied the dose accumulating into tumors by about 15-fold. And we've completely eliminated or decreased, let's say, by over 90% the kidney and salivary gland dose. So this is really a big, big breakthrough. This is how much radioactive dose goes into the tumors if you inject the same amount of activity. This is the new compound we have. This is the one that's coming into clinical trials next month. And this is the new optimized one. And you can see the blue bar compared to the orange bar is how much more radiation we're delivering to the tumor sites. And this is how the dose of radiation from the tumor to the kidneys, and this shows how much it improves. So almost a 10-fold improvement as well in the safety ratio of those compounds. So the hope is that we can push the dose much higher and drastically improve the efficacy. And we've seen this with mice, and I fully acknowledge that mice are tough critters and we can treat them with very, very large doses of various stuff. And we can get good tumor response. But this is a mouse that has a tumor, a prostate cancer. This is a human prostate cancer that we treat. And this is the number of mice that are surviving this long. So these are those that do not have the treatment between the blue and red lines. These are the ones that receive a dose of PSMA 617, which is 14 times the single dose that we would give to a patient. So the drug works, improves survival. And this is when we give the new compounds. When we give the same dose as the orange one, essentially there's long-term control for the cancer for all the mice, where most of the mice are actually cured. When we give a quarter dose, it still is much more potent. The survival is improved almost twofold compared to PSMA 617. So we're quite excited about those results. We're even more excited about what it means when we use Actinium 225. So we do not have yet these curves for survival. So we're just looking at the tumor shrinkage because those experiments are ongoing. So this is fresh off the press. We haven't published this. These are the compounds when we treat mice, and we can see with a single dose, very fantastic response. So it'll take longer to figure out at the lower doses whether some of those tumors are coming back, but we're seeing really good signals, at least in mice. But I don't want to get everybody's hopes up. This needs to go to human clinical trial. It will take time, but we really have, what we know is that PSMA 617, which is coming next, is a very effective treatment. There's a lot of trials about combination therapy to improve on this trial. But we can certainly see, you can expect to see upcoming trials coming to BC with these new therapeutic radioisotopes, probably late in 2021 as well as 2022. I'm already a year and a quarter behind the schedule I showed you. I was looking at that the last time where I showed this group 2 years ago. So things have a nasty tendency to take longer than we want or we anticipate them, and there's various reasons for that. But we're really excited to take this forward to the clinic. So I'll stop there. Maybe take the time for questions.
It's in my monthly cancer Zoom call.
We have a couple of questions, Dr. Benard. Is there a limitation on how RLT is used? Example, a bone, vascular lymph nodes.
Visceral.
Yeah, there is no limitation. So if for what we call visceral disease is disease that would affect the lung or liver, for example. Lymph nodes are when the cancer is spread to the regional nodes, but there's no limitation. So the radioligand therapy can be used for both bone and non-bone disease. So it works equally well. And this is the big advantage over radium-223, which only treats the bones, for example.
Next question I have before we get to Richard. Also, if applicable to lymph, does it matter if the lymph is on the heart or the lung?
No, it shouldn't give enough radiation to the particle range is not sufficient typically to irradiate the heart in significant quantities or in the lungs. So these are not contraindication to giving the treatment. The bones is a bit of a tougher situation because if the cancer is spread extensively in the bone marrow, which can happen late in the course of the disease, the bone marrow could be exposed. So there's some controversy about this. People tend, in Europe, for example, they like to use actinium when the bones are so involved. But I was talking to one of my colleagues at the Peter Mac in Melbourne, and he was saying he's treated people with extensive bone disease that were not eligible for the VISION trial, and they have responded well, and their bone marrow activity is actually improved because all the cancer cells were preventing the normal cells from forming bone marrow. But this is potentially a source of toxicity that even with the new compounds, we will not be completely able to prevent this secondary radiation of the bone marrow when it's highly infiltrated, which is why I strongly believe that it's going to be important to treat people probably earlier and not wait until the last minute as the last course of treatment with this approach.
Richard.
Hi, Dr. Benard, you give the best lectures in prostate cancer that I've heard in the 10, 11 years I've lived in the city, and you're doing, I think, superb work. I just want to say that at the outset. I have a bunch of questions, and I don't want to lose everybody, but I'll start with the simple ones that won't lose people. I get to hear a PSMA theranostic talk at least once or twice every year from various scientists. And they all show, of course, the Rtrlt's famous papers with the patients from 2016 or 2014. And I always ask this question, are those spectacular patients still alive?
Yeah, so the survival advantage so far, because we don't have, I don't know about this patient because they need to preserve the anonymity and so on. We do know that there have been reports of a couple of very long-term survivors with those treatments. So meaning that I suspect, to be honest, that this patient is still alive, the one that went down to 0.1. So there's reports of a couple of patients like that who've been what we would call in medicine extreme responders. So, you know, the kind of responses that we're so happy to see, but it's so rare that, you know, almost everything goes away. So there's reported examples of that. It's not the majority of people. So what we know from the studies is that the average survival advantage of getting the treatment is about 9 months with PSMA 617 when people are treated very, very late in the course of disease. And when they're treated with actinium, which are often even worse patients, the life expectancy advantage on average is about 15 months. So that means that there's people who, you know, might be shorter. Some people could be 3 years or 4 years. So I can only give you averages, but this is where we are, what we know. And they're not very well designed prospective studies. Like we'll know from the Vision trial a lot more in a few years. We'll know from the Australian studies as well, and we'll know from our own Canadian study what the impact on survival. But it takes time, because now, fortunately, men who develop advanced prostate cancer, the disease, it's much better than it was just a few years ago.
Yeah, I have two more questions. One is, one's a sort of a economic political question. I'll save it. But the other one is a sort of a biochemical one, and I don't want everybody to get lost in this. But your albumin binding compound, that's your AB compound. Can you tell us a little bit more about the molecular target and how that's what you envision is going on there?
Yeah, so what we've done is we've learned a lot by doing the diagnostic compounds on how to make the compound bind better to PSMA. But what we were, you know, you probably have a biochemistry background, I suspect, or if I recall correctly. What happens is that there's an accessory binding pocket in the receptor. So I'll just use a bit of jargon for Richard now. Sure. And we've added a hydrophobic component to the molecule. We've played with it until it binds very tightly to the protein. And but whenever we were doing this, the kidney and salivary gland option the binding kept going up as well. So we fiddled with the other part of the molecule to stop that binding. And we came on with a combination of being tenacious, persistent, trying a lot of things, and quite a bit of luck. Now we ended up compounds that still bound to PSMA, but no longer bound to the kidneys and salivary glands, at least in mice. So I don't know 100% for sure in patients. I'll know in a month, but at least we've been able to solve that, we think.
I love it.
Yeah. Go ahead.
I love the work. I so much want to see the results. And I'm happy if I can, some way as a patient, promote your work, let me know. But allow me to jump to this sort of touchy political economic one. So I think the last PSMA talk I heard was by Schaefer out of Triumph. And at the end, I asked the question, you know, which he didn't say. I said, come on, what is this? What is the actinium going to cost us? And he sort of skirted around the question. You may have even been at the same talk. And sort of blamed Novartis. Oh, well, if it's expensive, it isn't because of the actinium. But he never said how much the actinium is actually going to cost. So at this particular moment, what do you figure the actual baseline cost is going to be for one dose for an average patient for one treatment of actinium PSMA?
Well, I can tell you what it costs in the US. And in Canada, the problem is that when the drugs discuss price, they develop these confidential agreements, and in order to get discounts, they don't want other countries to know what discounts BC is getting, for example. And I don't want to find out, to be honest, because then I couldn't talk openly. So in the US, it's about the Lutetium, we know, is about $48,000 per cycle, and they receive 4 doses, similar to the prostate cancer, so $40,000 US per cycle. In Canada, typically, we pay the same price, but in Canadian dollars. So we pay lower our drugs. So I suspect that the PSMA 617 will be charged at about $40,000 per treatment. I do not know for sure. I'm not speaking from knowledge. This is speculation. Just want to make it clear with everybody.
So how do we as patients lobby the government, because they're not going to be happy to hear that, How are we going to get them to be prepared? How do we as patients work with you as a scientist to point out that this is going to be really expensive, but likely to be really good? We've got to get the public behind us to accept the fact that our medical care is getting more expensive. We need the PET scanners. They're really expensive. The one from UC Davis is elegant as hell, but going to be outrageously expensive. How do we get this word out that people are going to be willing to pay for this?
I think it's by advocacy, and you have to be careful as well, because sometimes the drug companies use patient support groups in order to pressure the government to accept a higher price. So there's a fine balance there, but it's advocacy for the importance of making this accessible and the value that is being brought by these treatments and your perspective on the values. So we're building the health economics part in our trial in part to be able to generate data on this, but it's not me or the government to decide what's an acceptable cost per quality adjusted life year. This is the language they use to estimate those costs. It is really the reflection of the public's value in this. You know, what is the value of investing there versus building roads and things? So the government has to make tough choices, doesn't have unlimited budgets, although you might think so after the past years, but typically they negotiate aggressively on the national level. So this is sometimes why it takes a little longer now to get drugs in Canada, because they'll say we won't make it available anywhere in Canada unless you get your price down. And typically they're able to get substantial discounts. For example, it might be 40 at the beginning, they might end up paying 25 or 30 per dose so they can treat more patients. The actinium will be even more expensive, and that's for sure. Because the isotope is very rare. But I think at the end of the day, in the era of complex, expensive biological targeted therapy, the scary part about prostate cancer is because it's so common. But I think you'll see that there'll be the typical balance between the need for care and the government's pressuring the company to make the cost affordable, and it'll come to some sense. And I suspect that you'll see within 3-4 years that this will be integrated in part of routine practice. The key problem is what to do in the meantime, and this is problematic for people who men who need access and can't get it. And what we're trying to do is to open up as many trials as we can to enable access. The Australians have been probably the best in the world to do this, and we're trying to learn from their experience. I'm trying to do the best. You know, I spend most of my weeks, my time trying to accelerate that process. There's a lot of resources being put into enabling this through trials as much as possible. And this is the way to make it available before.
Thank you very much.
Thank you, Richard.
I have a question.
One comment I might make to that is if we want more healthcare, I think we have to be prepared to pay more too. Anyway, there's another question from Ontario. Can Ontario residents join the BC clinical trial?
So the national trial, the Pluto trial, is opening up nationally. So this will open up, there's at least 13 centers who've expressed interest. 6 or 7 will probably be off the ground relatively quickly. So the hospitals in Montreal will be the CHUM and the Jewish General Hospital. In Hamilton, it's going to be the Hamilton Health Sciences Center. So I'm not sure if it's St. Joseph who will participate. London, Ontario will participate as well. And I strongly suspect both Sunnybrook and Princess Margaret as well will participate in that trial. We're working hard for Alberta as well. Calgary is quite keen to do this. Access to PET scan, PSMA PET scan is currently the problem in Alberta. So we're going to try to see if we can try to make it available for them in BC to get that going. But people would have to fly in BC in these COVID times. It's a little bit problematic to get the scan. Edmonton is working hard at developing the diagnostic tracers as well. So I have colleagues there. So we're hoping to really make this as widely available as possible.
I have a question.
So the next question I have here, just a moment. Oh, sorry, Michael, you had a question?
Yeah. Yes, I do. I'm currently in the Arrow trial at Princess Margaret in Toronto, and they're using a 131-1095. How does that compare with what you've been talking about? Do you know?
So the Arrow trial is the iodine 131 treatment for targeting PSMA. Is that correct?
Correct.
So this is a similar drug that binds to PSMA, but it uses a different radioactive isotope called iodine-131. So iodine-131 is extensively used to treat thyroid cancer. So it just uses a different radioactive isotope than the lutetium. So it's great that you're able to participate. We wanted to participate in BC, but by the time we got to them, they'd had enough sites. So it's another PSMA targeted radioligand therapy. And I can't tell you, is it going to be better or worse or similar to PSMA 617? We'll need to see the data once the trial is completed. I do not know at this stage, but it's a similar type of radioligand therapy that uses a therapeutic isotope that is proven effective for thyroid cancer. So it's a great trial to be on.
Thank you.
Next question. I think you mentioned this before, Dr. Benard. A lot of trials seem to be making a comparison with the use of chemotherapy. This would suggest that the patients in the trials had advanced cancer and had spread to their bones. Is that true?
That's correct. I think what happens is that by the time people develop androgen or what they call castration-resistant prostate cancer, the majority of situations, the cancer is spread to multiple locations. They may not always be in the bones. It could be in the lymph nodes as well as in the bones. Because of the trials were done after standard of care treatments were exhausted, so after chemotherapy, typically the cancer had time to spread to the bones. And I would think that most patients in the trial had advanced cancer that had spread to the bones. I think we'll see a different patient population when we do our comparison with chemotherapy because people will not have had chemotherapy. So it's likely that many will have had the cancer spread to the bone because that's often one of the sites where it spreads rapidly. But we see on the diagnostic scans a lot of people who only have lymph node disease as well. And because we can pick up that disease before and it was just not found on MRIs, on CT scans before. So I think we'll see disease earlier with less involvement in the bones in the new trials that are being planned.
Next question, interesting one. This gentleman from Toronto would like to know where he could get the lutetium treatment in Germany.
So there's multiple sites in Germany. There's hospitals, there's hospitals in Munich, there's hospitals in Heidelberg that I know of, Bad Burka. It's pretty easy to find different hospitals to do it. They do charge patients to get the treatment, and it's very hard because of travel restrictions now to get there to get treated. But there are a number of sites in Germany that offer non-German treatment, but they operate that a little bit like private practice. They will charge, and people have to be mindful of when they are getting the treatment, because they will treat, depending on the centers, they will treat many people and some with very early disease. And I personally do not think we have the evidence that it's the right time to treat them at this stage with those treatments when hormone therapy is very effective. So my advice in general, people, if they haven't been treated, if their cancer is responding to androgen deprivation therapy, meaning the hormone treatments, and if they're still responsive, that is still the best treatment to get based on everything we know, and it's not yet time for radioligand therapy. The radioligand therapy, I think, will come in when the hormones no longer work. But we are seeing more trials taking this earlier. So it wouldn't surprise me to see trials comparing it directly with hormone therapy quite early in the process, but we need to find out what's coming off those trials.
Next question is, which Montreal hospital will be participating in these trials?
So I think I already addressed that. There's the ones I am expecting in Montreal is the Jewish General and the CHUM, the Montreal University Hospital on the French side. I think Quebec City and Sherbrooke are also scheduled to participate. That's outside Montreal, though.
Let's see, one more. I've heard of India. I heard India also provides lutetium for much less money than Germany. Of course, not a place to go today. Is it available in India that you're aware of?
Yeah, I know of groups in India that do offer that in trials. I do not know specifically whether they take people for payment, for example, to treat them. That I'm not aware. It's a long ways to go, obviously, and not the time. But I know they have trials going on in India. They also have a good actinium trial going on in Africa, in South Africa. But I don't know if they take people from other countries, because in these clinical trials, often you kind of need to be able to follow and monitor people fairly closely.
Australia, Australia. Would there be people coming from Canada to go to Australia for treatment?
Yeah, there are some sites and there's private clinics. So Australia has both the public and private treatment centers, and I'm pretty sure in Australia there are opportunities to get this done privately through trials.
Any other questions? Anybody wants to ask any other questions? If they raise their hand, I should be able to identify them. I'm hoping to anyway. Anybody else?
Greg had a question.
Yeah, I actually typed them in there, but being metastatic with cancer in my bones, how do I know when to get treatment before it spreads to the marrow?
It's a good question. I think if it's metastatic to the bones and people are no longer responding to androgen deprivation therapy, then it's when people would start thinking about getting chemotherapy or participating in one of the radioligand therapy trials. So if you're responding still to ADT, then I think they would know from imaging, from bone scans or from PSMA PET scans, or they would know from your blood counts, that if they start dropping and so on, that could mean the marrow is starting to get affected a lot more. But if you're responding well, your PSA is responding well to the hormone treatment. First line, or whether it's the standard hormone treatments or whether it's enzalutamide or abiraterone. Currently, I think I would continue with those drugs unless it's as part of a trial. But if after this, then the PSA progresses, and for example, there's evidence of progression on the bone scan, then it's to start to look at the next stage. And your physician, your oncologist, and you are really the best to determine when is the right time. But I think I would say, I cannot give specific advice, but in general advice, I think to consider chemotherapy would be both a combination of symptoms, quality of life, and balancing this all out about when's the right time to start. For radioligand therapy, I think as soon as you're eligible to a trial, I would just ask, see if there's an opportunity to participate.
Yeah, okay, and that was another thing, is how to get into a trial, because I am on hormone therapy. It's great. I have undetectable PSA, but it's been 3 years now. So, it could start going up at any time. When it does start to go up, how to get into a trial. And also, with IAMI being built here, my other question is, why would we be paying these outrageous prices to some US company when we're producing it right here?
Yeah, so those are two questions, but the first question is a trial, is you talk to your oncologist or urologist and express your interest in participating in trials. And their responsibility is to look up for you and provide you opportunities. There's another site that's called clinicaltrials.org. It's a US maintained site where you can search for available clinical trials. So you can do the homework on your own, but typically your physician should be able to direct you, and they will be able to give you advice whether a trial is relevant or not for your particular situation. And the thing about ADT is it can work for 10 years as well. So the androgen deprivation can work for a long time. And if you were to get treatments too early, then something else better comes in. You might not be able to they might exclude people who've had radioactive treatments from participating. So I think my advice for people who are well controlled on ADT and they don't have severe side effects or heart problems is, just stay the course for now, because those treatments can be very effective. Second question is TRIUMPH. Yes. So Triumph is making it sporadically right now. So it's making one and one. I can get some once every quarter or so from them. I'm getting supplies from Germany, not the US currently, on a fairly reliable basis because they're producing it differently. The facilitated IMI is not yet up and running. So it will take still a bit of time before we can get a regular supply of lutetium from the new beamline and the new infrastructure being built there. I suspect at least a couple of years, if not a little longer than that. But then how Triumph will commercialize it and so on, there's a lot of questions still remain to be determined. There's a lot of demand. I could speak about it might be fun for another session about the supply of actinium, where it's coming from, because it's super interesting. Right now, we're living off nuclear waste from the nuclear bomb research done by the US and other sites in the 1940s. So it's really a rare supply. They're processing, they're hiring companies to clean up the nuclear waste, tons of material, and extracting tiny amounts of material that can be used to make actinium out of that. So there's a little bit of supply from there in Russia, Europe, US, and a little bit in Canada. But there's also the production beamlines at Triumph that can make some of it, and they're ramping up to do a lot more. But demand will outstrip capacity. It will take probably at least 3-4 years before we see some kind of balance. But when there's money to be made, there's private companies will invest in making this available. And I suspect that we'll see a lot of private investments to start going into this area. There's been billions in the past 2 years of money being invested in various ways to develop new drugs that use radioligand therapy. So things will change quickly now that the financial sector is looking at this as a way to make money.
Leo Fox, if you unmute yourself, you can ask your question. Leo Fox in Toronto.
Yeah, so my question, which I wrote, is why wouldn't you use this for fairly early stage? Like I'm Gleason 7, but I've had now I think 3 or maybe 4 PSMA PET scans, sort of opportunistically, a couple in Australia, a couple in Germany. And it just struck me during the last one, hey, why wouldn't you use this for treatment rather than just imaging, which is exactly what we're talking about here. So why wouldn't you use this as kind of prophylactic, preventative? Your early stage, okay, we do this, and you never get to the stage where you have to use ADT or radiation or surgery.
Yeah, it will happen. And there is a trial open in Australia to treat primary prostate cancer with this and see how effective it is. And so people will and are trying to study that part. Right now, the main disadvantage when you treat early, for example, if I get prostate cancer, I get surgery, it might take 10, 15 years before it comes back, or it might take 2 years. So it's very variable between people. But if it's 15 years and then it relapses, and I could have another 10 years with androgen deprivation therapy, then it might be 20 years from the time of diagnosis until the cancer spread and it's really bad disease. Could be longer, it could be shorter. But when we know that when people are treated by radioligand therapy with the current family of compounds, that the kidney function declines about 10% per year. So it can vary if you receive less dose. The kidneys might be 5, it might be a little bit higher. But the kidneys are affected. So if people have a long life expectancy when they receive the treatment, we could be causing problems to their kidney, needing dialysis or having problems down the road in 10 years. So currently, it's not a problem when you're treating disease that's advanced and the life expectancy is much shorter, because you don't worry about the kidneys. You want to keep the person alive and give that person individual a good quality of life. But if you treat earlier and earlier, then this becomes a much bigger problem. And the higher you want to boost the dose, let's say there's a dose escalation study at Cornell, they're trying to see how much can we push this up to treat the cancer and improve the response. And you can give a lot more, but the kidneys will be affected. And we still don't know enough to know how much can we safely give to the kidneys, but we have learned from the Lutetium experience and others that there's an upper limit. And it's different as well for a young person with completely healthy kidneys to somebody over 60, where if you look at the American population, about more than 50–60% of people have either hypertension or diabetes or both above the age of 60.
Wow.
And that damages the kidneys as well. So the kidneys are less resilient. So this is my big hope with what we developed in the lab, is that we can get rid of that problem. And I'm really, really nervous and excited about the prospects of being able to solve that problem. But I don't know for sure that it'll work in people. And then if you fix the problem of the kidney dose and the salivary gland dose, then that opens up, makes those things much more likely or possible.
Frank Peabody, you have a question?
I was just wondering about the salivary glands. Is there permanent damage? It seems to stop them working, but is it a permanent problem?
So with lutetium, it seems to recover in between doses. So if you give a dose and then you wait 6–7 weeks, people can get dry mouth, but it gets better after a few weeks. So it seems to be mild to moderate, but transient with lutetium. With actinium, people need to stop treatment, which means that it doesn't get better. And it seems to be affecting the salivary glands much more than lutetium. I think it's just an efficacy and a dose effect that we're seeing, really, that even with lutetium, when people measure the salivary flow, the glands are permanently affected. And we know it's the same because when we give radioactive iodine treatment for thyroid cancer, it accumulates in the salivary glands as well to some extent. And we know that people have decreased salivary flow. It's just that there's enough salivary flow to not be that noticeable for people. There's enough so they can get by. But when it gets more severe, symptoms become more severe. People develop dry eyes and dry mouth, causes problems eating and so on. It's a very difficult symptoms to live with. So I think for lutetium, at the doses are currently being given, it seems to be well tolerated, not a major issue. People receive the treatment, it recovers in between cycles, and it doesn't seem to affect their salivary gland function too much. With actinium, it seems to be a much bigger problem.
We have one more question from Barbara. Barbara, would you like to unmute yourself and ask your question?
Hello, thank you very much for this wonderful presentation. Really appreciated it. A question, I just wanted to know, so far, do we know any predictive factors as to the benefit of this kind of treatment? For example, does the Gleason score play a role?
So far, no, we do not. We don't have enough patients treated to parse out what are the variables that predict who will do well. The one thing we know, for example, is if they have a high uptake on the baseline scan, they're likely to respond better. Another thing we know, and they are doing this in Australia, is that the PSMA expression gets higher as the prostate cancer becomes more advanced or aggressive until a certain point, and this typically happens after people have had multiple rounds of chemo. The cancer cells start to change and it turns into almost a different kind of cancer that's much more aggressive. It's called the neuroendocrine small cell cancer. And those, things get bad when it turns into this. And the cancer cells start to lose PSMA expression when that happens. And it is, so going back to your question, what the Australians are doing is they do 2 PET scans, one with FDG, and we know that the FDG will be better in aggressive prostate cancers than less aggressive ones, and they do the PSMA scans. And if people have FDG avid disease that doesn't light up on PSMA scans, they've indicated that this was a sign that, a predictor of poor response. So they do not treat those patients. So that adds an additional PET scan. Now, we haven't done this in our study because we're treating earlier. We expect to see less of that. And if you add more tests, then essentially we wouldn't have been able to have the budget to do all of those PET scans plus in the trial. So it's also, we sometimes have to do financial, economic decisions based on that. But these are some of the examples of predictive factors, a discrepancy between an FDG PET scan and a PSMA scan with disease that's FDG positive, PSMA negative, then those patients are likely not to respond well because it indicates that some of the prostate cancers do not express the target. And then the uptake on the scan is predictive. But beyond that, I'm very interested in, I'd love to be able to address that question. It will require treating a lot of people and figuring out afterwards. The other question we're interested is looking is whether there's genetic markers, meaning is there a subtype or what makes somebody respond super well and somebody else not respond. And it could be that's in our gene. The ability to repair DNA damage could be at, causing the differences in response. And that could lead us to ways to improve that. So we're going to collect blood samples and analyze the DNA of the blood. At some point, we'll need to get a grant and funding to do this. But as part of our trial, we're going to collect at multiple time points blood samples to try to study that. Try to see if we can find good predictors of who's likely to respond and who's likely to not respond.
Thank you.
No, any further questions? I don't see any further questions.
I think there's Mr. Khan, Jamil.
Yeah, that's right. In fact, I just seem to remember during the presentation, something with the brachytherapy HDR patients who came for this trial. So my question is that during the trial, what is the success rate of those who have been treated after the brachytherapy HDR, and particularly intermediate, unfavorable risk?
Yeah, so what we know with HDR, with both LDR and HDR brachytherapy, is that when you look at populations of patients, it's very, very effective. So it's a great treatment. It has similar response rates than surgery, and HDR is thought for more advanced disease to be better than LDR for more aggressive prostate cancers. So the control rate, very few people will relapse after being treated. But we initially it was thought that these people would not relapse in the prostate gland because you deliver such a high dose of radiation. It seems that some people can relapse there, but overall, if you compare people with brachytherapy, surgery, or radiation therapy, the overall outcomes seem to be similarly good for most people in all the groups. The problem happens with the bad actors or the bad disease. I'm talking not the patients, but the cancers, of course. They can relapse with any treatment modality, and this is a kind of a very different question when it's coming back. It doesn't necessarily mean that the initial treatment was not effective, but it can also mean that it had already spread at the time that the patient was treated, but we couldn't find it or see it on any imaging scan.
And I guess it's for this reason that the brachytherapy is combined with the ADT. And in that situation, the chances, I guess, or the data might as well show the chances are very minimal.
That's correct. So most people respond very well and the cancer doesn't come back, but it's a small percentage of people in whom the treatment failed, and that happens with all the treatment modalities that then need to get additional treatments afterwards.
Thank you.
Another question here, Greg, could you unmute yourself? You can ask your question.
Yeah, just one more question. Between the, with cancer in the bones, the lutetium or actinium, I know the lutetium tends to destroy a little bit more of the healthy cells around it. So is there more of a chance that the lutetium is going to destroy bone marrow in somebody with cancer in the bones?
That is what some groups in Germany are assuming. So they will take somebody who has just a few bone metastases. No, it will not affect the bone marrow function. If people have very, very extensive widespread disease that's affecting almost all the bones in the bone marrow, then when they have access to actinium, they will reserve the actinium to these patients with the thinking that the normal bone marrow cells are less likely to be affected by this crossfire effect. But they haven't proven it. So we don't know. So the way to do it would be to take similar patients with say extensive bone marrow disease, and then you randomize them between lutetium and actinium. And is it really true that it's better or not? That's the only way we'll find out. But so it's possible that actinium is better when people have extensive bone marrow because the particle range is shorter and it's less likely to kill normal marrow cells, but it's not been proven. So this is really just a hypothesis. It does make sense, but I don't know for sure that it's true. And I think lutetium can be quite effective as well. And the reality is that actinium will probably not be as available as lutetium.
So if with just a few, with not so many metastases in the bones, then lutetium is fairly safe.
Yes. And I would probably go for that as a first-line treatment, because then there's always a possibility of receiving actinium as a rescue down the road as well.
Okay, thank you very much, and really excellent presentation. Thank you.
Thank you so much.
If there are no more further questions, Dr. Benard, I just want, on behalf of everybody, say what a wonderful presentation you've given us today, and it's most encouraging for us prostate cancer patients for the future. Thank you, thank you, thank you for your taking time to speak to our group. And I want to thank
Thank you very much.
Norm for pursuing your presence here today. It's been very rewarding to us all. So thank you, thank you, thank you again.
Perfect. Thank you so much for having me. It's a pleasure to be here, and you had great questions, so it makes it very interesting for me and fun to engage. So I really appreciate the warm welcome.